Hello again, Higher Biologist. Mr. Kinnear here with another video lesson for Higher Biology. We're looking today at photosynthesis, which is the second part of Key Area 3.1. So this is where it fits into the whole of Unit 3. So effectively it is plant growth and productivity, which you'll find on Scholar and on BBC Bitesize. But essentially we're looking at photosynthesis, which is obviously related to plant growth and related to productivity. So we've talked about photosynthesis over the numerous years you've been doing biology so far. So obviously green plants trapping energy of sunlight in chlorophyll. That then helps convert that light energy to chemical energy. And then we combine carbon dioxide and water to make glucose and oxygen. So raw materials, carbon dioxide and water, glucose and oxygen being the products. You learned some more detail last year in National 5, but funnily enough, now you're in Higher, we're obviously going to add a layer extra of detail, so we'll go through that today. We'll start the process by thinking about the light that is going towards a leaf. So that's obviously 100% light. Some of that, 12%, is reflected, and that's how you see objects. So whether it's a plant or a table or a chair in your room, and then you see those objects because light bounces off those objects, reflects off them, and goes into your eye, and then your eye changes that to electrical impulses, blah, blah, blah. So objects, including leaves, reflect light. Some of the light is actually transmitted through. So again, if you sit in the shade of a tree, not all the light is stopped by the leaves. So 5% actually goes through the leaves. There's about 83% which is absorbed by the leaf. Only 4% of it, however, is used for photosynthesis. You should know by now that the site of photosynthesis is the chloroplasts. They're found in green plants. They're large discus-shaped organelles. They are most abundant in palisade mesophyll cells, so those are the ones that you find towards the top of the leaf. We talked about that in National 5. A new bit of information, they're double membrane bound, and they have two internal regions, which we'll talk about as we go through. So the first region we're talking about is the granum. They are stacks of membrane-bound sacs. The sacs are called thylakoids, and they contain photosynthetic pigments. They're connected by intergranular lamellae, and that's where photolysis happens. And again, we'll go into the detail later on, but that's a name you should remember from National 5. The second one being the stroma, so that's a fluid-filled region that's full of enzymes. That's where carbon fixation happens, so that's the second stage of photosynthesis. And again, you will find starch grains present there. So they are temporary stores of the carbohydrates. So glucose, which is one of the products of photosynthesis, is going to get converted into starch immediately, and that allows again for that continuation of the high to low diffusion rates. So if I was to say photosynthetic pigment to you, you will probably talk about chlorophyll, because that's what we've talked about previously. But as with many things at higher level, we're finding that to be a slight half-truth, because chlorophyll, as we've described it before, actually consists of several different pigments. So chlorophyll A, chlorophyll B, xanthophyll and carotene, and all of them have the function of trapping light. And the different pigments absorb different wavelengths of light. So the total quantity of light absorbed is greater than it would be if we only had that one pigment, chlorophyll. So chlorophylls A and B are complex molecules. They contain the element magnesium. And if you do have a lack in magnesium, they normally have yellow leaves and they're said to be chlorotic. So in terms of those individual roles, chlorophyll A is said to be the primary photosynthetic pigment. It's the only one that can convert light energy into chemical energy. It does absorb light in the red and blue regions of the spectrum. And that's why we've talked about it as the sole pigment previously. But as I've just said, we've got chlorophyll B, carotene and xanthophyll, and they are accessory pigments. So what they can do is absorb different wavelengths of light that chlorophyll A can't, and then pass on the energy to chlorophyll A, thus broadening the absorption spectrum and therefore allowing more energy available to photosynthesis. And that can be shown in an absorption spectrum. So this is where you see the most amount of light being absorbed in the red and the blue areas of the visible light spectrum. As you can see, there's very little absorption happening in the green. And that's obviously why plants are seen as green, is because instead of absorbing that light, they are reflecting that light to your eyes, so that's why you see them. The light that is reflecting that area is normally done by carotene and xanthophyll, and obviously again, as I said, they pass on that energy to chlorophyll A to produce the products of photosynthesis. This second graph, however, probably more important, it's the action spectra. So we're now talking about the rate of photosynthesis dependent on the wavelength of light. So as you can see, if we expose leaves to red light, we get a good peak of photosynthesis. And again, down here, this end in the blue area, we're also getting a high rate of photosynthesis. And again, down here in the green, we're getting a lower rate of photosynthesis. And how can we come up with that? Well, you see, we expose different leaves to different wavelengths of light, and then you can measure things like the carbon dioxide absorbed, the oxygen that is produced, or the increase in dry mass. So obviously plant growth over time based on those different wavelengths of light that the plant is exposed to. So this is the action spectra. So here we have the two spectra superimposed on top of each other. So we've got the action spectra, that black line, and different absorption spectra of the different photosynthetic pigments in the colours. And as you can see, if we only had chlorophyll A, we wouldn't have this bump here in our action spectra because our chlorophyll A is here. And obviously the absorption of light is very low from chlorophyll A in this kind of area. But having chlorophyll B and the carotenoids increases the action spectra in this region, that it just wouldn't happen if you didn't have those extra pigments available to you in that leaf. So now that we have looked at the structure of chloroplasts in more detail and the photosynthetic pigments that are contained in there, we're now going to look at the chemistry of photosynthesis. And one thing you need to understand if you're not a chemistry student is oxidation is loss of electrons, which in metabolic pathways normally means the removal of hydrogen. And reduction is the gain of electrons, which in metabolic pathways is normally the addition of hydrogen. Oxidation normally releases energy, whereas reduction requires the addition of energy. And we've seen that already in metabolic pathways and respiration. And one thing to remember there, the oil rig term, so oxidation is loss, reduction is gain. So we're going to talk about that as we go through photosynthesis. In National 5, we already talked to you about two stages of photosynthesis. And if you remember, we've got photolysis or light dependent stage, and that's when we're using that light energy to split water into oxygen and hydrogen. And the second stage being carbon fixation or the Calvin cycle, and that's where carbon dioxide is reduced to make this carbohydrate, and commonly we'll refer to that as glucose. So I'll go through the wordy explanation of what happens in photolysis, then we'll follow it up with a diagram. So it occurs in the granum, so that's one of the new regions we've just heard about this week within the chloroplasts. And this is an extra layer of information. So light energy is absorbed by the photosynthetic pigments, and that excites electrons within those pigments. That energy is then used to split water into hydrogen and oxygen, and then also produce ATP in a similar way that we've talked about in the metabolic pathways, so driving production through the enzyme ATP synthase. Oxygen is then released as a byproduct. It's a waste product in this process for the plant, so it's released into the atmosphere. The hydrogen, however, is needed. And again, like we said in metabolic pathways, hydrogen just roaming about within the cell is really destructive. So it's accepted by a hydrogen acceptor, a coenzyme, which is then reduced by accepting hydrogen to form NADPH2. So this is a new one, similar to NAD from metabolic pathways, but NADP, NADPH2 in the reduced form. And both that NADPH2 and the ATP are required for the second stage carbon fixation. So here we have the same information in diagram form. So we've got light falling on a leaf, which is then absorbed by the photosynthetic pigments that produces high energy electrons. And those are used to drive ATP synthase, just like we see before, generating ATP from ADP and PI. We also then use those high energy electrons to split water into its component parts. So the first one being oxygen, which is released as a byproduct. The second one, hydrogen, and that is then picked up by our coenzyme NADP to form the reduced version NADPH2. Those two things, ATP and NADPH2, are the important parts that come from stage 1 that go off into stage 2. Oxygen, obviously another product, but it's a waste product, so it's released from the leaf. So carbon fixation is enzyme dependent. So each step requires an enzyme. It's therefore temperature dependent. It happens in the stroma, and although it doesn't need light, it can't happen without the ATP NADPH from photolysis from stage 1. And it involves the reduction of carbon dioxide to form that carbohydrate. The stage 2 carbon fixation, or the Calvin cycle, is something we talked about last year in National 5. But as I've said, what we're doing this year is adding on an extra layer of detail. So the next slide will be quite a bit of text, but also I'll put that into diagram form, so hopefully that will be a bit easier to understand. So carbon dioxide enters the stroma by diffusion. That is then fixed by an enzyme called Rubisco to a 5-carbon molecule called ribulose biphosphate, so known as RUBP. That then forms 3-phosphoglycerate. That 3-phosphoglycerate is then phosphorylated by ATP and combined with hydrogen from NADPH to form this glyceraldehyde 3-phosphate, or G3P. So phosphorylation, if you remember, we talked about in metabolism. So ATP is being split into ADP and PI, and that one of those PI is being added on to our 3-phosphoglycerate. We're also taking hydrogen from NADPH from stage 1 and adding that on. So that G3P can then be used to synthesize 6-carbon sugars, such as glucose. However, most of that G3P is then used to recycle into more RUBP using ATP from photolysis. So again, that's why we've got this cycle, and you'll see that in the next slide with a diagram, that we're talking about it in a cycle. So RUBP is a start and end of this cycle, just like you saw in respiration. So as I said, it's a Calvin cycle, a circle. We're starting with RUBP, the 5-carbon molecule, and we'll finish there as well. So RUBP is the carbon dioxide acceptor, and that's facilitated by the enzyme Rubisco. So it's Rubisco that joins carbon dioxide to RUBP to form 3-phosphoglycerate. That 3-phosphoglycerate is then quickly phosphorylated by ATP and combined with hydrogen that's been donated by NADPH. So those are the products of stage 1. So stage 2 Calvin cycle can't happen without the light-dependent stage photolysis happening. That then forms G3P, 2 molecules of a 3-carbon molecule, and that will return to RUBP through ATP turning into ADP. But we're talking about photosynthesis, we're talking about the production of sugars, so we also want to convert that into 6-carbon sugar molecules such as glucose. But you can also convert those G3 molecules into things like proteins, fats, nucleic acids, which are obviously important for plant growth as well. So in terms of the products of the Calvin cycle, the end products of photosynthesis, glucose, or a 6-carbon sugar molecule, can be used straight away for respiration. It can also be stored as starch, and obviously that is where we get a lot of our food products from plants storing their glucose from photosynthesis as starch. It can also be used to synthesize cellulose for cell walls. So again, the growth of a plant in terms of the products of cellulose. But as you saw from that diagram, there's also other pathways that can stem from photosynthesis. So producing proteins, fats, carbohydrates are also derived from photosynthesis through complex enzyme control pathways. So G3P being a precursor for fats and proteins. So that is how photosynthesis directly relates to plant growth, but obviously relates then to our previous lesson where we talked about food supply. And that's us today in our discussion about photosynthesis. We've talked about light being absorbed by different pigments within the granum of the chloroplasts. So not just chlorophyll, we're now talking about chlorophyll A, chlorophyll B, xanthophyll and carotene, which are all capable of absorbing different wavelengths of light. That then lent on to the absorption spectrum, which shows the amount of light being absorbed, and the action spectra, which shows the rate of photosynthesis, and that's compared versus different wavelengths of light. And lastly, we talked about the different stages of photosynthesis, photolysis, so the breakdown of water into hydrogen and oxygen, and carbon fixation, so a bit more detail on those two stages this year. Hopefully that has been helpful and informative. Obviously, if you have any problems, questions or queries, get in touch and I will speak to you again soon. Bye.